And so the thyroid gland, again, with our hypothalamic-pituitary axes, the one we just covered, ad nauseum is the HPA axis. So what we have with that is the hypothalamus secretes CRH, and that targets the pituitary, which releases ACTH in response. The ACTH targets the adrenal glands, and the adrenal glands release a panoply of hormones, including the famous cortisol, but also things like androgens and our other mineral corticoids as well. So again, many different hormones from the adrenal cortex. Now, with the thyroid gland, you still have the hypothalamic pituitary secretion, but now it's a different set of hormones. So remember that releasing hormones are always released from the hypothalamus to the pituitary. So with the HPT axis, what we have is the hypothalamus secretes a releasing hormone called TRH, or thyrotropin releasing hormone, and then the pituitary gland, that stimulates the pituitary gland to release TSH, and this is thyroid stimulating hormone, which stimulates the thyroid to make thyroid hormones. And again, the two main thyroid hormones are T3 and T4. So again, TRH stands for thyrotropin releasing hormone. Releasing hormones are always between the hypothalamus and pituitary, and this is TSH is the thyroid stimulating hormone. Now, where is the thyroid located? Well, here we have the thyroid cartilage, and actually it's kind of a slight misnomer. Notice that the thyroid cartilage isn't directly shielding the thyroid. It's actually located a little above it, and this is what you call the laryngeal prominence. And if you're a male, you can definitely see that, right? This is what's colloquially or commonly known as your Adam's apple. But yeah, you have your thyroid cartilage. So this is a useful landmark in determining, like if you go to an endocrinologist and they suspect you might have a thyroid gland problem, what they will do is feel your thyroid, and this is an important landmark to feel where your thyroid is. Again, you don't want to really press hard on that because you also have important blood vessels right here with your carotid artery and whatnot. But yeah, if they suspects there's something abnormal with it, if they can sometimes feel it through palpating your thyroid gland. And then this is what we have here. So inside of the thyroid gland has many of these structures we call follicles. And inside these follicles, we have the inside the follicle is called the follicle cavity. Sometimes in other textbooks, it's called the follicle lumen. And whenever you hear that term lumen in the body, and especially in anatomy and histology, lumen refers to the inside of some sort of like tubular spherical structure, some sort of round structure. So thing is that it's called a cavity, but usually when we think of cavity, we think of a hole, but it's more like a space, and this space is filled with something we call colloid. And it's a specific colloid substance we call colloid. It's mostly water, but also has a lot of protein and iodine, and the precursors or the starting materials for what will eventually become thyroid hormone. And there's also C cells, and these are parafollicular cells. There's another name for them. These actually secrete a different type of hormone called calcitonin. And if you want to know more about calcitonin, go back to, I believe it's in my skeletal system slides from PHYL 141, but that's a calcium regulating hormone. So follicular cells, these are the ones surrounding a follicle, but these C cells, notice that they're not in the follicle. They secrete a different type of hormone. But again, we're talking about thyroid hormone in this lecture. So we're focusing on these follicular cells surrounding the cavities or lumen. Now, what about these follicle cells? Well, what we have here are capillaries, and these are tiny blood vessels. And we have all these iodide ions being floating around in the plasma of your blood. So what happens with the follicle cells is that they have little ion pumps that bring in iodine. And as they bring in iodine, it starts to build up iodine, and these diffuse through these follicle cells, and then into this space containing that substance we call colloid in the follicle cavity. So all the ion or iodine is being pumped in and then moving toward the inside of this round structure. Now then you have this protein that made by the follicle cells called thyroglobulin, and this is a tyrosine rich. In other words, it has a lot of tyrosine molecule or residues in the polypeptide. So yes, it is polypeptide, it is a protein, and then this protein, along with the iodine, goes into the follicle cavity. And what happens is that all the tyrosines or most of the tyrosines, I should say, have iodine attached to them once they're in the follicle. So these two come together, you attach iodine to tyrosines located on thyroglobulin, but this all happens here. Then, the thing about thyroid hormones, there are two main active forms. So the thing is that these tyrosine, and this is an amino acid now, so remember that polypeptides are long chains of amino acids linked together. So these tyrosine amino acids, they undergo a process called iodination, and iodination literally adds iodine to the chemical structure of these tyrosine residues. Now, when you have these two tyrosines that are close by to or close to each other on thyroglobulin, then they can be linked chemically. So what happens is you link these two ring structures on tyrosine together, and now you have what will eventually become a thyroid hormone. So once you separate this whole thing from the entire thyroglobulin molecule, so if you have 4 iodine atoms, and what we had here before you link the tyrosines, notice that this one has 2 iodines, this one has 2 iodines, so basically they'll either have 2 or one of these iodines. So what happens is that link these two together, and now you have what was formerly 2 tyrosines. Now they're joined together in the same molecule, and they have 4 iodine atoms. So this is why, and this is what we call thyroxine, or for short T4. And why T4? Count them. 1, 2, 3, 4. Now there's another main thyroid hormone called T3. So how do you get T3? Well, in this case, this tyrosine is only getting 1 iodine atom attached. So with this, but what if it gets linked to another tyrosine that has 2 iodine atoms attached? So what's 1 plus 2? 1 plus 2 is 3. So this is what we call triiodothyronine, or T3 for short. So it all involves attaching iodine atoms to tyrosine and then linking pairs of tyrosine molecules together. Now, how does this relate to thyroid hormones? So we're almost making thyroid hormone, but not yet. It's still stuck to that protein backbone of thyroglobulin. So thyroglobulin with all the iodines attached and all these tyrosines paired up and linked. So what happens, or actually rewind it a bit. So further details about T3 and T4. So the interesting thing about the triiodothyronine is it's more active. And what do we mean by more active? So it actually binds better to the thyroid hormone receptor. So it actually causes more activity from that receptor versus T4. So it kind of fits better in that little pocket in thyroid hormone receptor that fits either T3 or T4. The thing is that it's less stable than T4. And if you have a chemistry background or know the term half-life, it has a shorter half-life. That means it falls apart, the chemical structure is less stable in the long term. So over time, you have less and less of this intact T3. Whereas T4 is more stable, so it has a longer half-life, but it's less active and less potent because it doesn't fit into the thyroid hormone receptor as well as T3. But its stability is why T4 is the more abundant, and it's actually the primary way of storing thyroid hormone in your bloodstream because, again, it's very stable. And the other advantage of having this mass amount or relative mass amounts of T4 is that your cells can easily pop off one of the iodines and convert to T3. So it can kind of make T3 as needed. So, and the other interesting thing about T3, this is a little advanced, but there are 2 forms of T3. It depends on whether the tyrosine that has 1 iodine is on top or on the bottom here. So there's a T3 in active form, but this reverse T3, notice that the one tyrosine that has 1 iodine is over here. For some reason, this does not fit into the thyroid hormone receptor, so therefore, even though it has 3 iodine atoms, it was made from 2 tyrosines, this doesn't work. This doesn't activate thyroid hormone or receptors. All right, so back to thyroglobulin. Anyway, so then what we have here is that thyroglobulin with all the iodine added, it encounters that organelle we call a lysosome. So a lysosome, I think a common descriptor is the garbage disposal of the cell, but the lysosome actually has a lot of enzymes that help to digest things like proteins. And in this case, it's going to digest the thyroglobulin that has all these linked tyrosines that have all the iodine atoms attached. So lysosomes will digest this, and when they do that, they kind of chop up these pairs of tyrosines that have the iodine added. So now you finally have T4 and T3 being released from the polypeptide backbone, and now these can diffuse from the follicular cells and then into the bloodstream. And then, yep, so this is how we have T4 and T3, and it binds to this thyroxine binding globulin. So again, thyroid hormone is not very hydrophilic. It's more on the hydrophobic side. That's why it needs to piggyback on these proteins that help transport it throughout your bloodstream.